My name is Shannon Chand. I'm the Crops Extension Specialist with Saskatchewan Agriculture in Swift Current, and I'll be the chair for today's event. CEUs are being offered for CCAs on this webinar. In order to get credit, you need to attend the entire event live, and there'll be QR codes at the end of the presentation that you need to use to collect credit. If you missed the code and have attended live, please email us afterwards, and we can work with you to get your credit. I also want to mention that the 2022 Crop Diagnostic School will be back in person this year and will be held on July 26th and 27th in Swift Current. Today's presentation will be followed with live Q&As with the speakers. The main hub for today will be your webinar screen, and we'll share relevant downloadable handouts for today. That is the agenda for the meeting and Saskatchewan Agriculture event and publication information. The entire session will be recorded, so if you miss anything or wish to revisit anything afterwards, you can. Recording links will be sent out later today in a follow-up email, and an evaluation will pop up at the end of the webinar this morning. Please ensure that you fill out the evaluation, as they help us determine what topics people want to hear about and helps us prioritize research needs for the future. We have a few housekeeping notes to keep in mind during the webinar. If anyone has questions during any of the presentations, you can type them in the question box in the bottom right-hand corner of your control panel. We'll try to get to as many questions as possible, but we may need to limit them due to time. Your question will remain anonymous and will only be sent to me. During the presentation, you'll be muted so that we can ensure there's no feedback that will interfere with the audio. So our first presenter this morning is Dr. Jeff Shano. Dr. Shano is a professor of soil fertility and a professional agronomist who works in the Department of Soil Science at the University of Saskatchewan. He holds the Saskatchewan Ministry of Agricultural Soil Nutrient Management Chair in the College of Agriculture and Bioresources and is a fellow of the Agricultural Institute of Canada. He was born in Saskatchewan, raised on the family farm near Central Butte, and completed his undergraduate and graduate degrees in the College of Agriculture at the U of S. He continues to be involved in farming with his wife, Lynn. So we'll start whenever you're ready, Jeff.
I think I'm ready to go, Shannon. Thank you very much. All right, great. I think I'll try and get rid of my screen on here. All right. Excellent. Yeah, thanks a lot, Shannon, and it's a real pleasure to be here this morning and to talk to you a little bit about a project that was recently completed by a graduate student, Melanie Reed, looking at nutrient dynamics in 2 pulse oilseed intercrops, chickpea flax and pea mustard, at 2 locations in southern Saskatchewan. And Melanie worked with myself and Dr. Diane Knight in her research project that she's just getting towards ready to defend her thesis, and also with Lana Shaw at the Southeast Research Farm. And also we had an ancillary site that we used in 2019 that Bill May had at Agriculture Agri-Food Canada Indian Head as well. So I'd like to thank all the folks who have worked together to bring this project to I think a successful completion. And it's great to have the opportunity to share some of our findings with you folks today. No, I'm just seeing if I can advance here. For some reason, I'm having some problem. There we go. Sorry about that, folks. So I guess it's a bit of background here, and starting out as the first speaker today to I guess warm folks up a little bit. Of course, intercropping is growing 2 crops together in the same season, and intercropping, particularly with pulse crops. In other words, grain legumes has some potential benefits that I think have come to light in the research work that I'm going to talk to you folks about this morning. And one of those is lowering the input costs. And that really is largely a function of the ability of the legume partner to fix its own nitrogen. And also, as I'll show you and revealed in this project I'm going to talk about, that those legumes can also supply nitrogen to the partner in that intercropping arrangement as it grows throughout the season. And the other thing about legumes, folks, is that they're really great scavengers of phosphorus and other nutrients. They can alter their rhizosphere, acidify the pH, that will bring phosphorus into solution. Legumes are a highly mycorrhizal crop as well, which helps in that plant being able to obtain phosphorus and other nutrients like potassium and sulfur from the soil. So they do a real good job of bringing nutrient into the active cycling in the soil plant system. So in this research work, we had a couple of objectives. We wanted to take a look at the yields and in particular the nutrient uptake by the crops. And also, because we have legumes in this intercrop arrangements, that were the two intercrop arrangements we were looking at, biological nitrogen fixation contribution in the intercrop pulse oilseed systems versus monoculture. And in the pulse oilseed intercrops, we had both mixed row and alternate row comparisons that we're able to make to their equivalent monoculture crops. And a lot of the emphasis on this was on nutrient cycling, nutrient dynamics. So we were really interested on the effect that those intercrops were having on nutrient dynamics in the root zone. So the hypothesis that Melanie really was aiming to test in her work were that there would be greater yield, nitrogen and phosphorus uptake within a pulse oilseed intercrop system than when those crops are grown as a monoculture. Following on along some of the previous studies that have shown this kind of synergy, also that biological nitrogen fixation will be enhanced in the intercrop systems, and that nutrient supply will be greater in the root zone of the intercrops than in the monoculture crops. So in this study that was completed in 2019 and 2020, which were relatively dry years at both of the sites, the intercrop combinations that we evaluated were Kabuli chickpea, brown flax, and dry yellow pea and white, or what sometimes people also refer to as yellow mustard, that's Synapsis alba. And those were the intercrop combinations that we worked with, because these, based on previous work that has been done in Saskatchewan, and particularly by Lana Shaw at the Southeast Research Farm, and by others as well, Agriculture Canada, showed these to be a couple of particularly promising combinations. So we had two sites in this. We wanted to take a look at performance and nutrient dynamics in different soil types, contrasting soil types and climatic conditions. So at Central Butte, we had a brown chernozem, a loamy textured brown chernozem. And then at the Southeast Research Farm at Redvers, a loamy textured black chernozem. And the seeding arrangements that we compared in this study, monocrop pulse crop, a monocrop oil seed, and then the intercrop with an arrangement in alternate rows, and then intercrop with the two partners in mixed rows. And this was done as a typical or traditional small plot RCBD randomized split block design. We have 8 treatments in this in the study, 4 replicates of each. So we had 32 plots per site, and the row spacing that we're using in this study at all sites in all years, a 25 centimeter row spacing. In terms of what we grew and our seeding rates, and these seeding rates were based on past experience and recommendations by Lana, based on her research work at the past research work at the Southeast Research Farm. Our yellow pea was seeded at 180 kilograms per hectare per row in the mix arrangement, and 209 kilograms per hectare in the row in mono and alternate. Our Adonti white mustard was 4 kilograms per hectare in row in the mix, and 8 kilograms per hectare in the row in mono and alternate arrangements. And then for our chickpea flax combination intercrop, we had CDC Orion, it's a Kabuli chickpea, at 200 kilograms per hectare in the row in the mix, and 231 kilograms per hectare per row in the mono and alternate. And then our flax was CDC glass, and it was 12 kilograms per hectare seeding rate in row in the mix, and 56 kilograms per hectare in row in the mono and alternate intercrop arrangements. And again, these seeding rates were based on what was found to work well in the past, based on previous intercropping work here in Saskatchewan. Now, in this study, we seeded everything without any fertilizer at all. And the reason why was we didn't want to have any fertilizer masking any of the effects of the crops themselves on nutrient availability. So no fertilizer was used, but the legumes were inoculated with the appropriate rhizobium species to maximize fixation. We did apply pre-emergent herbicide, ethylfluralin, in the pea mustard plots, and sulfentrazone in the chickpea flax plots. And overall, across the 4 site years, I think we had pretty good weed control in our plots with that strategy. To assess the fate of fixed nitrogen and to assess nitrogen fixation in general by the legumes in the partner, we used N15 labeled ammonium nitrate that we applied in microplots within the main plots. So what we did in methodology, we took samples in the spring. In season, we used PRS, plant root simulator probes, placed in the root zone in between to track the supply rates of available nitrogen and phosphorus. We also took samples out of the root zone and in between the rows during the growing season to measure soluble nitrogen and carbon, which is microbial substrate. And in the case of nitrogen, a significant portion of that soluble nitrogen is plant available. We also removed roots and did staining and counts for arbuscular mycorrhizal fungal colonization. And finally, at harvest, we took 2 1-meter rows in which we used to measure the grain and straw yield and measure the concentration of nitrogen and phosphorus to determine N and P uptake. And in our N15 microplots, we also measured grain and straw yield of the legume, the percent nitrogen derived from the air, from fixation, and the nitrogen uptake. And then at the end of the season, we took post-harvest soil samples as well to look at the effect on residual nutrients. So that was our methodology strategy. That's a photo there of Melanie out collecting root zone rhizosphere soil during the growing season, shaking it away from the roots of the plants that were removed. And those plants that were removed, chickpea, flax, pea, and mustard plants, also were brought back to the lab, and the roots stained and counted for mycorrhizal colonization on the roots. And also during the growing season, we had the PRS probes, plant root simulator probes. There we can see them in the root zone. During the growing season, you can see just the tops of the probes there. They were placed into the soil, and every, I think, 2 weeks, we replaced them with another set of probes so that we could look at the supply of available nitrogen and phosphorus in the root zone over the growing season. So what did we find in this work? And this covers 2019-2020 growing season. And I will say at the sites in both years, we encountered some dry conditions, particularly dry conditions in early spring at the Central Butte site in 2019, which caused some problems with germination and emergence, and also some dry conditions later in the season in the 2020 season, particularly at our Redverse site. Before I go on, I just want to remind folks of this important concept that is used in intercropping called land equivalent ratio, or LER. And what it simply is it's the ratio of, for example, the yield or the nutrient uptake in the intercrop divided by the yield or nutrient uptake of the monocrop on an equivalent land area of 1, and then added on to the second intercrop in the mix, the yield divided by the yield of that same crop grown as a monocrop. And when we do that calculation, if we get an LER value greater than 1, that indicates a benefit from intercropping. And basically greater efficiency if that LER value is greater than 1. Greater efficiency, and I guess we might say LER greater than 1 indicates a synergy. So moving on through to the parameters that we looked at, the first one, yield, we did see evidence of greater efficiency in producing yield from the land area in those pulse oilseed intercrops by LER values that we obtained that were greater than 1. In agreement with previous studies that have been done looking at yield and in intercrop and calculating LER values. So at the Redvers here in this example, the pea mustard grain yield LER, and at both Redvers and Central Butte, we can see their values greater than 1. And you can see the yields of the individual crops in those mixes as well, the pea in green and the mustard in yellow. We also calculated LER values for nitrogen and phosphorus uptake. And again, we saw LER values for nitrogen, phosphorus uptake in above ground material, grain and straw above 1, indicating a synergy in these legume oilseed intercrops. Showing there in this example and chickpea flax mixture at Central Butte, we have the grain nitrogen LER values and the grain phosphorus LER uptake LER values on the right-hand side. And those LER values were higher in 2019 than in 2020. 2019 was a really quite dry spring at the Central Butte site. There were some germination issues, and we kind of got some variation there in emergence, but overall, seeing those LER values for nutrient uptake above 1, and also observed in the pea mustard combinations as well at the 2 sites over the 2 years. We calculated what's called the nutrient utilization efficiency, which is the yield produced per unit of nutrient uptake. So kilograms of grain yield produced per kilogram of uptake of nitrogen in the above ground material. And we can see there, looking at those graphs, in this case using the Redvers chickpea as an example, nutrient utilization efficiency for nitrogen on the left-hand side, nutrient utilization efficiency for phosphorus on the right-hand side. Those alternate and mixed row intercrop systems are producing significantly higher nutrient utilization efficiency for both those nutrients compared to the monocrop system. So we're seeing there a benefit there that nutrient is producing more yield, probably for a variety of reasons, not just nutritional, but also just some of the other benefits that come from that intercropping system as well. Now, when we looked at nitrogen fixation, we did not see any difference in the percentage of nitrogen in the legume partner derived from fixation. That is the percent NDFA. Some other previous studies have shown enhancement in percent NDFA in the legume partner in intercropping systems because of the partner oilseed or cereal drawing down the nitrate level, the available nitrogen level in the soil, increasing the percent NDFA. But we didn't see that in this particular study. But one of the things that we did see that some of the other studies have not observed or were not able to document was that the biologically fixed nitrogen was being transferred to the oilseed partner in the intercrop. So when we look at those graphs there below, we can see that, for the percent NDFA, there really is no significant difference in the mono or the alternate row or the mixed row arrangement. But in particular, percent nitrogen transfer, we were seeing anywhere from 15% to upwards of 40%, according to our calculations, of nitrogen being transferred from the legume to the non-legume partner, the oilseed, during the growing season. And this was also observed at the Indian Head site that we assessed in 2019 as well, a similar pattern. And again, in both the pea mustard and the chickpea flax intercrop systems. Now, providing support for this transfer, we did observe a higher root zone soluble nitrogen, that is inorganic and organic N, contained in the root zone, higher root zone soluble N in the root zone of those pulse crops alone and in the intercrop was likely a contributor to that intercrop synergy. And is shown there in those graphs for pea mustard at our 2 sites with the pea soluble organic and inorganic N shown there in green in the mono and the intercrop system. We can see there that soluble nitrogen is higher. And following along with that, in agreement with that and providing support for that transfer, we also saw an enhanced root zone supply of nitrate using our PRS probes there that would also be a contributor to that intercrop nitrogen uptake efficiency. Shown there for the chickpea flax, the supply rates of available nitrogen in the legume, the chickpea, shown there in the orange in the mono, and the alternate arrangement being higher than what we see there in our oilseed partner. And again, showing that what that would contribute then to that N uptake in terms of seeing that higher LER value, that LER value greater than 1. When we calculated the potential removal of nitrogen in the cropping systems, we did observe that the intercrop oilseeds had a lower potential removal of nitrogen from the cropping system than the monocrop oilseeds. So we can see there in terms of the effect on the nitrogen balance, the potential removal of nitrogen from the system. Looking at those two graphs, we see that both the flax in either mixed row or alternate row arrangement, and the mustard in the mixed row or alternate arrangement has lower potential removal than when the flax or the mustard is grown alone in a monoculture. And part of that is attributable to this transfer of that fixed nitrogen to that flax or mustard during the growing season as they grow together out there in the field. Overall, I will say, folks, in this study, we didn't see a lot of difference in the parameters we looked at between the mixed row arrangements and the alternate row arrangements. Although there was a tendency sometimes for those mixed row arrangements to have some superior performance over the alternate rows, particularly in things like the nitrogen transfer. And finally, looking at the percent root colonization by the arbuscular mycorrhizal fungi, which we did through staining and root counts, we had a little bit of issues with this because of COVID, because we ended up bringing samples in from the field that ended up being stored in the laboratory for longer than would be desirable. So we had some challenges, but overall we saw pretty high colonization, as you can see, in general by the AM fungi on all of those crops that we were evaluating at our different sites over the 2 years. Not a lot of differences there, as you can see, between the monocrop and the intercrop, either alternate and mixed row. We did see in 2019 in redvers and the flax, there was a slight but statistically significantly higher colonization in our mixed row versus our monocrop, but overall not a lot of differences there. And probably, I think, if we were to do this again, we probably would also apply a molecular technique as well to take a look at AMF populations. The staining technique had some limitations in terms of its ability to reveal differences. I think one of the important things is that in terms of phosphorus, we do see and did see a greater crop uptake and removal of phosphorus when we had the intercrops compared to the monocrops. And when we see that higher LER value, that greater uptake of phosphorus over the same land area in an intercrop system, we do realize that down the road, that's going to mean that we're going to need to be putting back in more phosphorus to replace that. And that showed up, I think, in Central Butte 2020. Available phosphate supply rates with the PRS probes were lower in the arrangements where we had the mustard or the pea together, growing together compared to a monocrop type system, where we're seeing a greater drawdown, a greater consumption where we have the two crops growing together. So to conclude, folks, overall, those two pulse oilseed intercropping systems we looked at over those 2 years certainly showed evidence of synergy, of increased nutrient efficiency that I think can to a large extent be attributed to that observed in-season transfer of soluble nitrogen from the legume to the oilseed partner. These were pretty nitrogen deficient soils, no fertilizer added. In terms of phosphorus, we didn't see any strong evidence for enhancement of P availability in the rhizosphere when growing together. There were some trends there evident, but we do see greater overall phosphorus uptake and removal associated with the intercrop. And that's something we need to think about when we're thinking about maintaining soil phosphorus fertility in these systems over the long run by replacing what we're taking out. Overall, as I said before, no large consistent differences between the alternate and the mixed row intercropping arrangements observed. And also to remind folks, this study was conducted with some periods of drought stress at both sites and both years. And I think it would be interesting to take a look at these effects under better moisture conditions. So with that, I'd like to thank you for the opportunity to participate. That's Melanie there, and also Ryan Hangs out working out in the field at one of our sites. I think that might be our Central Butte our research site there. And special thanks to our funding agencies, the Agriculture Development Fund and Western Grains Research Foundation, also to the Saskatchewan Flax Development Commission, who provided a scholarship to Melanie Reid. Thanks to Ryan and the rest of the group out there who helped with carrying out this project over the last 2 years. So with that, I will stop, Shannon, and turn it back over to you. And I think we've got about 5 minutes for questions. Thank you.
Yep, we're right on time. Thank you very much, Jeff, for a great presentation. Just as a reminder, if you have any questions, please enter them into the chat box on the bottom of your control panel. So the first question, were there any differences in economic returns?
We didn't calculate the economic returns on this. I mean, I think you see, if you compared it to the monocropping system, just in terms of yield, we did see some pretty impressive synergy there in terms of our LER values there that were above 1. Now you also have to take into consideration the seeding rate aspect of that and the cost of the seed. But I think, you know, from looking at a yield per unit land area basis, I think there would be a significant economic benefit that if we calculated that out for the 2 years. In this study, we were mainly honing in on nutrients, nutrient availability, and nutrient cycling.
No, that's great. Thank you. What was the fertilization rate of the monocrops in the study?
There was no fertilizer added at all to any of the crops.
Okay, thank you. Any thoughts or experiments where nitrogen was banded below the non-legume in alternate rows? Maybe it could benefit picking up the nitrogen before the pulse recognizes it or brings it in.
Yeah, I'm not sure how much benefit you would get from adding the nitrogen fertilizer to the pulse, because the general effect of adding especially high rates of fertilizer nitrogen to pulse is a reduction in fixation. And although you may be separating that nitrogen initially by placing it in a separate band, that nitrogen is mobile. So it's going to get through and it's going to have that inhibitory effect on the nitrogen that's derived from fixation. And that's really the big benefit from that pulse, that legume in that intercropping system that we see, is that ability to bring that nitrogen in from the outside through fixation in those nodules, and then transfer it, shoot it over to the partner in the intercrop combination.
Oh, that's great. Thank you. Were any ending grain quality parameters measured?
Not in this study. No, we just did grain yield. And you know, I have to say as well is that our yield measurements were based on 2 1-meter rows in our plots. And you know, that's probably not the best assessment of the yield, but we do that because we want to very carefully sample and get not only the grain but the above ground straw as well, so that we can measure the nutrient recovery in both.
Okay, thank you. Did you notice or record insect pressure in the intercrops and disease pressure? And also, awesome presentation.
Great. Yeah, thanks. I guess from the disease pressure standpoint, we didn't measure that directly, but Dr. Michelle Hubbard from AAFC Swift Current went out and took a look at the plots out there. I think 1 year for sure, 2019, and that was a year where there was some issues with foliar disease on the chickpea. I believe it was ascochyta, and I think there was an observation that there was less of an issue with the disease in the chickpea in the intercrop system compared to the monocrop system. And especially it seemed that what was able to suppress it was the alternate row arrangement. Now at Central Butte site, I know, and I'm a little bit more familiar with that one, we did have some issues with chickpea disease, foliar disease in 2020, and that reduced the competitive ability of the chickpea. We got it under control through the application of a fungicide. But you know, when it comes to competitiveness of the two partners, it's certainly influenced by things like disease pressure and also the initial soil fertility. We found that the pulse was a more competitive partner when the available phosphorus level in the soil was low. And maybe under those conditions, you would be able to reduce the seeding rate of the pulse relative to the oilseed partner. So those types of competitive interactive effects do have some ramifications in management, whether it's, you know, disease control or even the things like the seeding rate. Oh, I can't hear you now, Shannon.
Sorry, I knew I was going to forget to unmute myself at least once, so hopefully I've got it over with. Well, I have one more question. There are more questions, but we'll probably move on after this one. Will there be further discussion on practical aspects of intercropping, like seeding depth or combine settings, from this study, or the studies you've done?
From the studies I've done, I suppose what we've got now, again, the emphasis was on nutrient dynamics, but the data is all there, and it'll all be there in Melanie's thesis, and hopefully ultimately in a publication in short order, so that folks can, and you know, I would be able to, you know, apply some economics to that, do some calculations there, so with based on what we got for the yield. Unfortunately, you know, we didn't do quality parameters. We measured the nitrogen concentration, so we do have the protein content in the grain, and that's another consideration as well.
No, for sure. And thank you very much, Jeff, for all the great information and the
Good morning. Can you share, Shannon, can you hear me okay?
Yeah, no, we can hear you and we can see your PowerPoint.
Okay, sound good. Okay, thanks, Shannon and Ali, for inviting me to present today. Thanks, everybody, to join the seminar today. So this one was I presented maybe 1 or 2 times. I really appreciate Dr. Jeff Shannon's presentation because many of his introductions and background information really helped my presentation as well. So this is slightly different from Dr. Jeff Shannon's presentation. It's more towards forage utilization and not the green crop. So this is started in 2016. We're using a different forage feed mixture with the cereal crops for green feed productions. Before I go through, I want to share with you our project team members. We have MSc students, Amara and Alex. Both of them has graduated, and I think Amara is working in Saskatchewan, and Alex went back to US. She's originally from Texas. In the team, we have Dr. Tom Workington from Plant Science, and Greg Penner from Animal and Poultry Science, and Dr. Diana Knight from Soil Science. We also have collaborators from AFC, Dr. Avasa, Brett Molson from Melfort, and Rosemary from Swift Current. So original idea was really starting as an adopt project by Trevor and many other Saskatchewan forage specialists. And we had more questions, and this project initiated in around 2015. I also appreciate the funding from Saskatchewan Ministry of Agriculture through ADF, Saskatchewan Forage Seed Development Commissions, SaskPows, and Saskatchewan Forage Network for graduate student assistance. Today's presentation, I want to talk 3 parts. First experiment is really forage biomass yield and how quality changes when we change in the mixture of the different species. We also want to talk about nitrogen fixations. And this was really interesting because Dr. Jeff Shannon mentioned about adding some nitrogen, whether that really reduce the benefit of having pea crop in the mixtures. And we also did some, based on this, we moved on to the next stage and we did some animal performance trials, and I give you some update on that one as well. So a number of questions for using pea cereal mixtures. Provincial recommendation is seeding pea crop 100%. So it's really high seeding rate and reduce cereal crop to 25 or 50%. The reason of high is pea crop, it's not very competitive, especially in a drier area. However, this also brings some of the issues such as it's very expensive seed, and potentially drying is a concern. Lodging can be an issue as well. And previously, 4010 is older pea varieties used quite commonly, and maybe even nowadays as well. However, CDC Horizon is newer variety, which is supposed to be more lodging resistant. Another question is, based on this, can we reduce seeding rate to 50-50? So we potentially get benefit from cereal crop for more biomass, and some benefit from nitrogen fixation and protein from pea crops. And there are a number of many questions around it because it's going to increase the competitions of the cereals to the pea crop, and maybe we don't really see too much pea component in the mixtures. And also, it's probably very common to producers. They want to add more nitrogens, not too much, but maybe only 50 pounds per acres, and increase the yield of cereal crop. But they also worry about this kind of adding small amount of nitrogen increase the cost and did not get the benefit because reducing nitrogen fixation of a pea crop. So based on these questions, we did a study in 2016 and 2017. It's identical trials in 2 years at 3 sites. We used 4 replicate randomized complete block designs, Melfort in a black soil zone, Saskatoon dark brown, and Swift Current brown soil zone. We have 12 different mixtures. We used 100% pea crop and 30% cereal as compared to 50-50. We also used nitrogen fertilizer. So we added some nitrogen fertilizer because we are trying to understand benefit of nitrogen and adding nitrogen. So we try to find the site which has a very low amount of nitrogen. So all 3 sites we tested soil, and we found nitrogen level is quite low in certain areas. So we used 2 years. So this is a nitrogen amount in the soil. So we really didn't add 60 kilogram hectare of nitrogen. Rather, we added, based on the soil test, we added up to 60 kilogram of nitrogen in nitrogen treated plots. Before I go through, I give you some overview of the crops we used. 4010 is a really old variety of pea crops. It's a full leaf, and seed coat color is different from CDC Horizon. It's a newer type. It's more lodging resistant. Flower color are different, but the major difference is a leaf type. This is a semi-leafless. Leaf is really reduced. I give you some difference in the next slide. We also used Maverick forage barley. The unique thing about Maverick forage barley is it's smooth, so it's really more suitable for feeding. We used CDC forage oat CDC haymaker. It's a very wide leaves and slightly taller than regular oat type, and also based on the breeder Dr. Aaron Beatty and Brian Rosnagel, they mentioned forage oat potentially more drought tolerance than regular oat varieties. So CDC Horizon, if you look at this, this leaf is really reduced. So the tendrils, and this called tendrils, and this is 4010. You see a full leaf. And then when we showing these pictures, people always ask whether this reduced leaf potentially reduce the protein levels. And we in the monoculture, when we're working with Dr. Tom Markington, we tested a number of times, and somehow we really don't see that much difference between these 2 types. So you can see this is a few trials of this CDC Horizon P versus 4010. So this is earlier stage of lodging, but if you really for 4010 wait until harvest, it's really flat, and that's a major disadvantage of this variety. So seeding ratio, we used for soil crops, CDC Horizon, and also 40-10. It's a seeding ratio. When we use 100% seeding rate, it's a pound per acre. You can see from here, CDC Horizon is maybe slightly a smaller seed. We targeted similar plant density, and CDC Maverick barley and CDC oat, it's this much, but target it's this for each site. So all 3 sites we use the exact same seeding rate. The difference here is we reduced cereal 0 to 1/3, and then this is a regular, and this is a 50-50. So it's a 50% of this seeding rate, and this is 50% of this seeding rate. So we have 12 different mixtures, all 3 sites. So this Melford 2017, CDC Horizon and 100%, and barley 30%. I give you some pictures. So CDC Horizon 50-50 with oat. And one thing over the experiment we found is oat is way more competitive than barley, and we also observed that one, and also the data in a legume component also shows that one as well. So this is another site pictures from Saskatoon. We have CDC Horizon and barley, and 40-10. So if you look at from these pictures, 40-10 is really looking really good, more leafier and larger plants too. The only concern is its lodging. So forage harvest stages, regular forage harvest for silage is barley harvested for soft dough stage, oat harvested for late milk stages. But based on Dr. Greg Penners and some other faculty members in animal science, they recommended harvesting later for grain feed. So we harvested in a hard dough stage of barley, which is maybe slightly later than, yeah, I personally probably prefer slightly earlier, but oat, it's a soft dough stages. And in 2 years, 2016 and 2017, 2016 was a good year. It's a normal year. We got a very uniform distribution of rainfall and had a good stand for all 3 sites. 2017 was a drier year, especially in swift current area. So we come to a result. So if you look at lodging, it's quite clear. As expected, we see a quite severe lodging in 40-10 when it grows in a monoculture stand. CDC Horizon was pretty good in overall. However, when we adding a cereals as expected, we reduce lodging, especially adding barley and oat in 100%. It's probably slightly better than 50-50. However, still we see some issue with 40-10P, and this was quite significant in Melford area. So forage yield, was 3 sites was different. So I'm going to talk with you about by each site. In the Melford area, what we found is adding nitrogen, it's actually increased forage yield. So I probably have to give you, so this is a monoculture stand. This is a barley or oat with a CDC Horizon P, and this is barley or oat with a 40-10P, and my next few slides are same. So we see the increase because of adding a small amount of nitrogen, and this may be because Melford area was more moist compared to Saskatoon and Swift Current, and maybe soil type is different, soil organic matter is different. So all this probably work together and see some benefit. And this is Saskatoon. When we adding a nitrogen fertilizer, we really didn't see the benefit. Another thing is this is a monoculture pea crops versus this is a monoculture barley or oat. When we mixing them together, we didn't really see yield advantage of this mixture over their monoculture cereal crop. But if you look at this, the component, the green is pea crops in the component. So it's if you have 100%, you see more when you reduce to 50%, you're less. Overall, this component of pea crops for 4010 is slightly better. The reason you can see here better is we harvested by hands for this determination. So lodging really, it's not a big deal. So we harvest everything and we can determine the amount of pea crop in the mixtures. So there is not really huge difference between which pea crop you use. Yield was almost same. Maybe this one slightly better because of oat and lodging, etc. in Saskatoon. The sweet currant is very similar to Saskatoon. We didn't really see a benefit of adding small amount of nitrogen. We yield-wise, mixture is almost same as this monocrop, but in some instances, for example, oat, we see some reductions, and this was quite clear for 4010 compared to the CDC Horizon P. And I really think this is maybe because of harvesting. The 4010, if we grow it in a monoculture and we harvest in the proper time before lodging, we probably see more 4010 than the CDC Horizon. And see, here's the same thing. The difficulty we faced in this experiment is the lodging of a 4010. It's really flat and later in the season. So I give you some forage quality, protein concentrations, and protein yield. So I didn't really give you too many statistical analysis. I give you a mean of all of them. So the first, this is a pea crops, and on average we got 13.8% of protein. This is based on dry matter basis, so it's about 14% of dry matter is protein. This two zero crop, because we harvested slightly late, this probably really showing here is our protein level compared to regular grain feed. We probably expect around 9 or 10, but this is slightly low. And this is the average of 3 sites. And this is 9.9% when we averaging all this 130 ratio of pea crop and cereals, including oat and barley. You can see this is a this is barley. This is barley. This is oat. Oat is slightly low, and this is Horizon P and 50-50 compared to the 100. So you see when we reduce P component as expected, we see some reduction of concentrations. But the interesting thing here I want to show you is by acres or by land areas. Based on dry matter yield and then concentration of these proteins, we calculated how many kilogram of protein yield we're receiving per hectares. If you look at this monoculture, although we know yield is higher compared to or similar to mixtures, mixtures really improving the protein yield per acre basis. And we know this monoculture pea crop is lower. However, you can see compared to oat and barley, which is way higher yield, you probably see more protein yield. And I really think this is a value of having another component pea crops in mixture of zero crop. So starch is another important thing. The starch concentration of our experiment was lower than silage barley reported in Western Canada. It's kind of 210 kilogram of concentration of barley silage in Western Canada on average. Ours is lower. However, we looked at other data when we have P0 mixtures. This is not in Western Canada. We found the range is very similar. The concentration wheat is around 83 to 149 kilogram, and ours is somewhat clear range. So we expected more zero crop, probably see more advantage of starch, and we didn't really see. If you look at this, this really zero crop probably has a very similar trend in overall. So barley is higher, and you can see it overall barley, and here's a barley too. But this is really numerical because if you look at the latter groups, they are almost similar. So another thing we looked at was acid detergent fiber. Acid detergent fiber is It's an indicator of digestibility of the feed. And what we thought was this adding pea crop probably reduce this ADF, but we really didn't see any of this. If you look at here, barley has a lower one compared to oat, which probably is always the case. And here is a similar trend because of this barley. Mixture with barley is lower than oat. But we really didn't see the benefit of having pea crop in the mixtures. They are almost determined by zero crop. One of the reason for this is maybe because the component or the percentage of pea crop in our mixtures is relatively small. It was smaller than I expected. So this is just now I added this slide. We have done so many different analysis, and the whole the MSc thesis is really big. So I don't want to overwhelm you, but when I listened to Jeff's presentation and he mentioned about this land equivalent ratios, I brought it up here as well. So this is based on biomass. So it's intercropping, whether intercropping benefit over monocrop. We also looked at land equivalent ratio for fertilizer, whether adding fertilizer has a benefit, and whether the site has advantage. So if you look at this value is larger than 1, it means it's advantage of this using intercropping. And if you look at all of them, and they are all higher than 1, so that really means intercropping is better based on this land equivalent ratio. Fertilizer, it's interesting because adding fertilizer versus non-adding, there's a difference. Without adding fertilizer is actually better based on this ratio. And the site is different. Saskatoon somehow was one of the best, and then the other two sites was very similar. So in summary, pea barley or pea oat produce higher forage yield than monoculture pea crop, which is expected, but it was very similar to monoculture of barley or oat, so there's no yield advantage. Reduction of seeding ratio from 130 to 50-50, increased biomass yield, at least numerically. So it's not really huge increase, but there's some increase because of increase of a zero component and reduction of pea crop component. Starch concentration was similar or slightly lower for pea cereal mixtures compared to monoculture of the zero crop. And ADF was similar in most cases, but it was lower in some intercrops than oat monocrop crops. And we expected that, but we didn't see everywhere, or we didn't see in all mixtures. So lodging of 4010P reduced when we including in the mixtures, which is probably expected, but the 40 CDC Horizon was quite lodging resistant variety. So I also briefly mentioned this biological nitrogen fixation of pea crops. So we adding a small amount of nitrogen, whether we losing the benefit of nitrogen fixations. For this experiment, we used a nitrogen isotope, very similar methodology mentioned by Jeff's presentations. We used barley and oat as a non-nitrogen fixing crops. So in this experiment, what we did was, this is one site. All three sites has a very similar design. So in this half of this 12 treatment, there's no nitrogen or no nitrogen added. And this green area is nitrogen added. And we seeded quite wider rows of barley all around. So we try to reduce moving of nitrogen fertilizer to the adjacent plots. So pea crop was inoculated using granular inoculant before we seeding all these trials. Some of plot areas we used very similar technique mentioned by Jeff. So nitrogen isotope diluted and watered, and then we marked off them together. We also tested whether there is nitrogen fixing going on. So we dug some plants from all sites, and we really see this. So this is pink color, and we also see some nodule in the pea root system. So nitrogen fixation probably is actively going on. So I just simply put amount of nitrogen fixed by each site. Technically, probably I couldn't see this. So it's nitrogen derived from air. So these two Saskatoon is the first one, Melfort and Swift Current. So the other is same thing. So this is CDC Horizon P and 4010P. This is also different ratios. So this is based on really a site. Swift Current is a drier site, we expected. And I also checked, is there any report? I didn't see Saskatoon or Melfort area report, but in Swift Current in the past, P monoculture actually fixed around 69. So ours is lower. There's a number of reasons, because we harvested as a green feed. We didn't really wait into the seed mature and give them enough time to grow. So that's one of the reasons. Another thing is in the Swift Current, we also see some drier years. So this is 2-year average. Maybe one of the dry year reduced this nitrogen fixed amount. So overall, we can see reductions in the mixtures. This is mainly because of we see less pea crops, and really that automatically converted to lower amount of nitrogen fixation. Because of time, I won't go through all the results, but I give you a few results. I found it's very interesting. So application of nitrogen fertilizer reduced nitrogen fixation rate at Melfort and Swift Current in both years, in 2016 and 17. We didn't see in Saskatoon. So this is really site specific. If you look at their regional nitrogen levels, they are very similar, but there's moisture restrictions and time of rainfall. All this really affect. So we did see some nitrogen reductions in two sites. Nitrogen fixation rate was greater for pea grown with some mixtures. This was the background given by Jeff's presentation, was really great. The idea is maybe the cereal crop keep using this nitrogen, and so nitrogen in the soil level is much lower than the monoculture pea crop level, and so maybe it's improved nitrogen fixation rate. So we really see this, and then also this site, when we have measuring this rate in the mixture stand, we see the fixation rate was higher, but we again, the number of pea plants in the mixture is lower, so overall fixation amount is lower. So I give you a very brief about the trial we did. So we did many small plot experiment. We got some result, and we moved on to next stage, and we did a large area animal trials. So we used the 50-50 ratio of the mixtures we used, the CDC Horizon P and Maverick Barley 50, Horizon P and Haymaker Oat. We didn't use 40-10. The main thing was the lodging. It's a quite concern for harvesting. So each crops, there's no experimental design. We seeded 2 acres each, and then we harvested them. So this is kind of agronomic management. I want to show you only a few things. One thing about pea cereal mixture is harvesting the swashing time, or whether they can dry fast enough. So off them harvest in August 4th in 2018. So we build off them in 13. So it's around 9 days. And oat was harvested slightly late, and maybe another 10 days. But we have some rain during this period. But the interesting thing is pea crop. We were really worried about pea crops. However, this drying time is pretty good. It is a final moisture level slightly higher, but still we didn't see any molding in the final trial. So the drying time is really determined not by the species that much, rather it's climatic conditions. If we have good 10 days of sunny temperature with some wind, maybe we don't have any issue with drying. So when we're doing a large experiment, it's way different from small plot trials. And we ended up, we didn't really get 10% of or 15% of pig component in the dry matter basis. We only get less than 2%. This really messed up the whole experiment, animal feeding trials. Because of this, we used this monoculture pea crops. Then we blended more artificially. There's no pea crop, so then we added to 15% of pea crop based on dry matters, and based on 30%, and this is the same thing. So Alex has done a huge amount of work in animal trials, blood testing, and all different things, but I will only give you a result on average daily gain and dry matter intake. So this is only 25 days of trial, so you probably have to aware this is very short-term project and only use 6 heifers for Latin square design. So every daily gain, when we know pea crop in the barley, with increased pea crop level, you see some benefit of increasing for barley. However, this wasn't really clear on oat. And dry matter intake was quite consistent if compared to no pea crops, 15% and 30%. There's somewhat interesting trend, but overall you see compared to no pea crop, dry matter intake was increased in both barley and oat. So this is probably another advantage of the using pea crop in the feeding system. So some other things based on this, we did some economic value analysis. So nitrogen, adding a nitrogen was beneficial at Melford site. You can add nitrogen and you get benefit from higher yield. However, this wasn't really seen in Saskatoon and Swift Current. Cereal crop is generally cheaper than pea crops. Adding pea crops in 2016 and 17 kind of cost, seed cost, we see cereal is way cheaper, and we using a cereal mixture is also cheaper than using just pea crops. And the barley seems better than oat as well. I didn't really show here. Another thing we thought was in the larger areas, we seeded in everything in the mixed rows. Maybe alternative row, maybe the better options, reducing the competition from cereal to pea crops. And however, this nitrogen is really site specific. Nitrogen little or non-nitrogen, depending on soil nitrogen level. If it's really high, ours is very low, and we still see some difference. And moisture probably very important for benefit of this. And another thing is harvest stage. For our experiment, we harvest slightly late, and then we found the good weather conditions and drying wasn't a concern. However, we also see much lower protein value and somewhat higher fiber concentration too. So this kind of thing is also need to consider. With that, thank you very much. If you have any question, I'm more than happy to answer.
Thank you, Bill, for that great presentation. We just have time for one question. For seeding, was there different depths for the peas versus the cereals, or were they all kind of mixed together at one depth?
Yeah, that's very good questions. For our small plot trials, we mixed all of them and then seeded all together in the same depths of the cereal crops. So it's somewhat shallow than regular seeding depths for pea crops. However, in the larger seeding trials, we actually seeded in 2 different depths, but we really don't know why. But the emergence of pea crop in that trial was was way less than we expected. So that's one of the reasons we didn't see enough pea component in the mixture. Another thing in that trials, we got a new area, and the area was very, very sandy. I always think maybe that is another reason for low percentage of pea crop emergence.
Thanks a lot, Shannon. Can you hear me okay?
Yes, we can hear you and we can see your PowerPoint.
Okay. Thanks a lot. First of all, thanks a lot for the invitation to share the research that we have been doing on intercropping at Swift Current since 2015. Just a brief acknowledgement of the different people that have been involved in our organic research program. Some of them are no longer with us, some have retired, and others have moved to other research centers, and there will be new collaborators joining us as well. So as you probably know, legumes are very important in sustainable crop production, and they are the main source of soil nitrogen in organic systems. don't know why I can't advance it. Okay. Here. Okay, so from the we have been working on a climate change roadmap, and in the draft that we already have in place, legumes are listed as being important to achieve significant reductions in nitrous oxide emissions and therefore to mitigate climate change. However, as we all know, there are some disadvantages of growing sol legumes, especially in organic and low input systems. They are negatively affected by the environment, either very dry or very wet. The most commonly grown legumes in the western prairies are susceptible to root rot, Fusarium and Aphanomyces, and as you know, root rot decreases in addition to impacts that it has on productivity. It also decreases nodulation and therefore nitrogen fixation. And there is no effective chemical control of root rot, which of course is of significance for all production systems. And this is just some pictures showing root rot in different legumes. So there has been an increase of root rot in legumes in space and time, and data that Chatterton from the Lethbridge station has published indicates that there was as much root rot in the dry 2017 as in the wet 2016 across the prairies. So Fusarium diseases, including root rot in legumes, were not very important years ago when a crop fallow sequence was the predominant practice. But then we had changes in agronomic practices, more pulses, short rotations, reduction in tillage, and increased use of synthetic inputs. There has also been impacts of climate change. And unfortunately, there hasn't been much progress in breeding for root rot resistance in legumes. So a few years ago, we realized that we needed to investigate other practices such as intercropping and cover crops for their many benefits, including the possible control of legume root diseases. And again, from the same climate change roadmap, intercropping and cover cropping has been included as alternative production systems that could mitigate climate change. So our intercropping research and the organic management in the brown salt zone was initiated. When it was initiated, there wasn't, and there still isn't much information on the benefits of intercrops to organic production in semi-arid areas. And so since then, we have been looking not just at weeds and yield, which is a portion, we feel that is a portion of the whole picture on intercropping, but have also been examining other variables in addition to the following sol and grain crop. And this approach has allowed us to understand different intercrop combinations and their multiple short and long-term impacts, positive and negative, a little bit better. And this is what I intend to show you, at least some of the highlights from this research. So the objectives of our organic intercropping research, the first three objectives are similar to those that other people have in relation to their intercropping work. Look at the nitrogen benefit from the legumes in intercrops, determine whether they can reduce weed biomass and density, and also determine the biomass, grain yield, and quality of the crops that are being intercropped. In addition, we investigated whether intercrops could reduce legume root rot. And finally, we also looked at the sol crop grown the following year, which in our case was durum wheat. So we looked at the whole combo, sol nutrient, weeds, biomass, grain yield, and quality, and diseases. So my presentation is basically divided into three parts. The first part are results from what we call phase one, which are the actual intercrops. And this encompasses two projects, one from 2016 to 19. That was our first intercropping project. And then a new one started in 2021, although there was a preliminary trial in 19. And this has to be postponed from 2020 to 21 due to COVID restrictions at Agriculture Canada. So the second portion would be the phase 2, which is the following fall durum wheat, and the third portion will be crop diseases and fungal communities in both phases 1 and 2. Phase 1 being intercrops and phase 2 being the following fall crop. I just wanted to acknowledge the funders of these projects. The first one was funded by WGRF, SAS Wheat, and SAS Pulse, and the current one is being funded by WGRF and POGA, the Oat Growers Association. And there have been several articles published on our work. This is one of them from the GrowPro magazine, and there's a few others, and they can all be downloaded from the Sask Organics website. So just I wanted to go quickly. I'm sure you're all familiar with the kind of environment that we have had in the last few years, and there has been a lot of variability in environmental conditions. We went from a very wet situation in 2016, where our field trials were flooded, to the start of a very dry cycle in 2017, and more or less it has been quite dry from then until now. And 2021 was basically the driest year that we have had in Current. And 2020 doesn't really count to us because, as I said, we weren't able to have any field trials. So in terms of the intercrops and their respective monocrops, phase 1, all our intercrops including a nitrogen fixing crop, a legume, and the non-legumes combined with the legumes were selected based on their competitive ability. So we had rye, oat, mustard, and flax, from the more competitive to the least competitive. The ones that I'm going to be talking about today are lentil and yellow mustard, and field pea and oat. For oat, we only used Oravina, which is a new cultivar developed specifically for organic production. We seeded in the first project, we seeded all intercrops in mixed rows, and each intercrop had different ratios of each of the companion crops. So this is just a brief summary of the different combinations that we had. We went from the legumes. We had the combinations that went from the fold rate of lentil, which was the same as the monocrop, to 50%. For pea, we use a higher rate because of the companion crop being oat, which is more competitive. So we went from full rate to 75%. And in terms of the non-legumes, we had mustard went from a full rate comparable to the monocrop to 50%. And for oat, we use a lower rate starting at 25% to 75%. And in addition, we also had forage pea manure, and we used 40-10 for that, and in all years, and it was used as a check, and they were all followed, as I indicated before, by a durum wheat crop. And we used a randomized complete block design with 4 reps each year. And this is just quickly indicating the seeding rates that we use for each of the crops, and usually our seeding rate is higher than for conventional crops because our operations are all organic. So just a couple of pictures on lentil and mustard. This is from the first round of intercrops, field pea and oat. And this is a picture from 2021. And in 2021, as I indicated before, we had different combinations, and I'll be describing them in a minute. And this is a picture of alternate rows of lentil and barley, 50% each. So in regards to the first round of intercropping, I'm not going to present any data, but I will be referring to those treatments when I talk about the following durum crop. So I'll be presenting some data from the current project which started in 2021. So we decided to use a wider range of seeding densities in each combination, including lower ratios of the non-legume crops and higher ratios of the legumes. And we used the same combination of field pea oat that we used before, but we introduced lentil with barley and also chickling vetch and mustard. But I will only be talking about the first two combinations in this presentation. And we decided to seed it in mixed and alternate rows. And again, all intercrops will be followed by a durum wheat. And of course, we still don't have any data on that. That will come in 2022 for this new project. So we also added other objectives in addition to measuring mycorrhizal fungi. One of our colleagues is going to determine inter and intraspecific competition and under various seeding ratios, and the objective is also to develop growth and nutrient uptake models and determine the optimum ratios for intercrops compared to their respective monocrops. And this data won't be available for a while. So this is just, again, some quick pictures. This was taken in 2019, P at full rate and oat 25. Then this is a picture when we decrease the ratio of oats. I don't have one with 5% oats, but it was pretty similar, and because of the oats' ability to compensate, which I will show in a minute. And this picture shows a higher rate of P, higher than the full rate that we use on the monocrop, so 125%, and oat remaining at 15. So these are the ratios that we used in 2021. So we started with a higher rate of lentil, going down to 50% for the alternate rows, and for barley we went from 5% to about 25% and 50% for the alternate. And whereas for pea oat, we also went from a higher rate than the full rate for the monocrops to about 50% for the alternate, and for oat we went from 5% to about 25% and 50% for the alternate row. So it's a little bit different than the first round, in that we decreased the ratios of the non-legumes and increased use a wider range of seeding rates for the legumes, but also increased them. So just to remind you how dry 2021 was. In terms of, I thought I would start with weeds. This is more or less the composition of the weeds that we observed in 2021. There were almost no perennials, and most of the weeds were red root pigweed, lamb's quarters, and wild buckwheat. So in terms of the wheat biomass, as expected and as observed before, the wheat biomass in the legume monocrops was the highest, or among the highest, and especially for the pea oat intercropping, it was significantly higher than in any of the other treatments, whereas in the lentil barley, it was higher, significantly higher in some cases, but not others. and the lowest wheat biomass were observed in both cases. They were observed in the alternate rows, which were not very different from, they were not significantly different from the barley monocrop in this case. And similarly, in the pea oat, the amount of wheat biomass was not significantly different from the oat monocrops. So this is where the lowest amount of wheats were observed. This table here shows the total biomass and total grain yield, and so this combines both the legume and the non-legume. And as you can see, the lentil and pea had the lowest total biomass and grain yield. And it was not in the case of lentil barley, it was not very significantly significant different from the intercrops, whereas in pea oat it was. And the lowest grain yield there wasn't much difference in biomass, but the lowest grain yield was observed with the lowest rate of the non-legume, here with 5% barley and here with 5% oat. So basically what indicates is that the ratio of the non-legume is what is determining the total grain yield. And in regards to the alternate rows, there was significantly, in some cases, higher yield and mostly yield, but also biomass in the alternate rows than in the mixed rows. And for pea oat, this was observed in the for total grain yield, but not for biomass, that the alternate rows provided a higher grain yield than the mixed rows. So in terms of the harvest index, and for those of you who might not familiar, this is the ratio basically of grain to biomass. And for lentil, there wasn't much difference between the intercrops and the monocrops. It was a little bit different. It was a little bit lower, sorry, but not significantly different. Whereas for field pea, it was in all cases, it was the harvest index of pea in the intercrops was lower than for the field pea monocrop. In order to help us understand what was going on better, we calculated actual to expected crop biomass and grain yield. And basically what we did was to, this was based on the values observed in the monocrops. So basically we multiplied the grain yield of each monocrop by the sowing ratio of the crop in each intercrop. So, and what we observed, and this is for lentil and barley, here we have crop biomass, and based on the ratio, for example, here 15%, we calculated the expected value of the barley, and whether it was higher or lower. And the same thing, we did the same thing for grain yield. So in all cases, we found that barley had a higher yield and biomass than was expected based on the seeding ratio. However, for lentil, it was lower in all cases. The biomass and the yield was lower than expected based on the seeding ratio, and particularly for grain yield, which, as you can see, was much lower than for biomass. So going into pea oat, this is the same calculation as we did for lentil and barley, and the increase in biomass and in grain yield compared to, based on the expected ratio, was higher than for barley. quite significantly higher in some cases, whereas for P, it was also significantly much lower than for lentil. So P decreased quite a lot when intercropped with oat based on their expected ratio. And also in regards to P, we found that that decrease was lower in the alternate rows than in the intercrop, in the mixed rows. And that has to do with the fact, this is a picture from 2021, it has to do with the fact that in alternate rows, there was lodging of the peas because they weren't able to climb onto the oat plants as we observed in the mixed rows. And so this is a close-up picture of the field pea climbing onto the oat, which seems to have helped grain yield significantly. So the land equivalent ratio was defined before by one of the previous speakers. So the issue that we had in terms of decreasing grain yield due to our environment was reflected in the LER value. Here you can see that for grain yield, it was never above 1. And for both the lentil, barley, and pea oat, it was always lower than 1. However, if we go back to crop biomass, we can see, and I indicated those in red, that the LER value was higher than 1. In lentil barley, it was the case when the ratio of barley was 25%, and also in the alternate rows when barley and lentil was present at 50%. But it was even better for pea oat. In all cases, the LER values were quite high, and compared to what we have been observing in the past anyway. And it was the lowest here when oat was present at 5%, and higher when it was present at higher levels. And among the highest LER values were observed in the alternate rows. And this is again mostly due to the oat ratios and not so much the P and the presence of the P. So we also looked at, by the way, we haven't looked at protein yet, but in the past there wasn't, we didn't find a significant effect on protein in terms of 1,000 kernel weight. In both cases, in barley and oat, the kernel weight that we saw in the intercrops was higher, although in the case of barley, it was not significantly higher, but tended to be higher. In the case of oat, the difference was a little bit greater. And for the previous intercropping project in 2017 and 18, this difference was significant in all cases. The intercrop oat, regardless of the ratio, it always had a greater 1,000 kernel weight than the oat monocrop. And as I said, protein, there was no difference in protein. So just a few highlights from this phase 2, which we have identified from the 2 projects. This is what other people have found as well, so it's kind of confirmation of what has been observed by other studies. The non-legume crops were always the most competitive, and among the mustard, oat, and barley were more competitive than flax. We did grow flax in the first project. And as I indicated, the higher the ratio of the non-legumes, the greater the impact on their companion legumes. And both crop biomass and grain yield, especially in the pea oat intercrop, was determined mostly by the ratio of the non-legume crop, which is in the case of pea oat, was oat. And as you saw a few minutes ago, um, the oat and barley were able to compensate for low seeding rates, especially in oat in 2021, and it was the same in oat for oat and mustard in 17 and 18, although I didn't show the data. So in all cases, the non-legumes were able to compensate for low seeding rates, and oat was the best in that regard. So under very dry conditions, legumes grew poorly, especially in the intercrops. And field pea was more competitive than lentil in 2017, and we also had soybean in one of the intercrops. It was the least competitive. And in 2021, unfortunately, field pea was relatively similar to lentil. It was not much better than lentil, but in 17 and 18, it was more competitive. So what are the pros and cons of intercropping under organic conditions in our environment, and also comparing mixed and alternate rows? We found that there was a lower wheat biomass and density in the intercrops and in their respective legume monocrops, and this was especially so in pea oat, where the differences were significant. And we also found lower wheat growth in the alternate versus mixed rows in 2021. We also found lower than expected grain yield of pea, shorter plants when intercropped with oat in alternate rows. That was due to lodging, and the grain weight of the non-legumes was higher, or tended to be higher, but not significant when intercropped with field pea and lentil than in their monocrops. And there was also, we also observed an insurance against failure of the legumes under very wet conditions. As I indicated before, our trials were flooded in 2016. The legumes didn't make it, but the non-legumes grew well, and we got a good harvest out of them. So what are the impacts of the environmental conditions? And to summarize them, the harvest index due to dry weather tended to be lower in the intercrops than in the monocrops. And in 2021, it was significantly lower in most intercropped pea in both mixed and alternate rows than in its monocrops. However, lower harvest index in the intercrops compared to the monocrops might also be attributed to crop breeding for maximum performance in monoculture, which is something that we usually tend to forget. And there's a recent paper by Chen et al. Published in 2021, where they explore this issue, the need to breed crops for intercropping if we want to pursue that agronomic practice. So as I indicated, there was the higher LER for crop biomass, suggests a high potential for intercrops under drought conditions. But however, on the other hand, the lower LER for grain yield, which was never above 1% and barely made it to close to 1, reflects drought conditions in the years in which these trials were conducted. So it is expected that under drought conditions, crops are not able to convert assimilates into grain, or there is not enough salt water for grain filling, and that would explain the differences in LER values. It's also expected that in dry environments, lower plant density than what we have used would improve grain yield. However, under organic management, high plant densities are needed. So as I indicated at the beginning, there isn't much information on the benefits of intercropping to organic crop production in semi-arid regions. So far, based on the results from our trials, we identified that under drought conditions, intercrops enhanced vegetative growth, and so it has the potential to successfully produce high amounts of crop biomass. However, if dry conditions persist throughout the summer, which has been the case, especially in 2021, this benefit might not fully transfer to grain yield. So again, it all depends on weather and whether dry conditions occur throughout the summer or not. So going into part two, the following soil during wheat, and this is for 18 and 19, and all the data that I'm going to present is basically a mean of both years. And so I'm going to be talking about weeds, crop growth, and grain yield of during wheat, and we use Transcend. So here we have wheat density in the during wheat after the intercrops here on the left were grown. And the difference in wheat density between the legume monocrop and the intercrops was quite significant. And so that indicates that the wheat seed bank was lower after growing intercrops than monocrops. And here among the lowest levels were observed in forage pea manure and also in some of the pea oat intercrops and oat monocrop, which resulted to be the most competitive and the ones that had the lowest impact on the following durum crop. So here is the some of the crop growth data, height, crop biomass, and grain yield. Again, this is all data after a year of intercrops here on the left. And as you can see here, biomass and yield were, well, first of all, they were highest after forage pea manure, which was not very different from a durum wheat grown after an oat monocrop. And in some cases, it was higher than in the intercrops, and but for yield, it was relatively similar, but higher than in the pea monocrop. On the other hand, the crop growth, in particular plant height and yield, was significantly lower in durum wheat grown after mustard and in the intercrops where mustard was present at a high ratio. The higher the ratio, the greater the effect, I guess. And it was similar to biomass in that the growth of durum was lower in the mustard and in mustard intercrops than in the lentil, than after the lentil monocrop, and of course it was in some cases also lower intercrops and oat monocrop. So what we did a contrast between mustard and oat, the growth of durum wheat after this monocrops, and also after the mean of the lentil mustard intercrops and the pea oat intercrops. And what we found was that in both cases, the contrast was significant, highly significant actually. So in all cases, having durum wheat grown after a mustard or an intercrop of mustard with lentil had a significantly lower biomass yield and plant height than after oat or intercrop of pea and oat. So, and this was related to the soil nitrogen level, which we measure in the spring after growing intercrops. So this was measured right after we seeded the durum wheat crop. And as you can see here, and there was actually no difference in nitrogen in the 0 to 15 centimeter layer, but in the 15 to 30 and 30 to 60, this difference between the nitrogen level after a mustard crop than after an oat crop and after a lentil mustard intercrop and pea oat was quite significant. And as you can see, it was basically double after oat and pea oat than mustard and lentil mustard. There was no difference for other nutrients, and it was just nitrogen that was quite where there was quite a significant difference. So the carried over of intercrop effects to at least the following year, and I'm saying that at least the following year because that's all we have been looked at, was the positive aspect was in terms of weed control, especially after field pea oat and with high oat ratios where there was a lower seed bank. So this is a very positive result from the intercrops. The negative, one of the negatives was a lower soil nitrogen level during crop growth and grain yield after mustard, and it's intercrops at high ratios. So that was, and I will come back to this in a minute. So those are the one positive and one negative impacts of intercrops on the following derm. So before I go back to that data, I just wanted to go over the evaluation of diseases and fungal populations in the intercrops and the following during wheat crop. So as most of you are aware, likely fusarium diseases are usually a complex, and we have them affecting many crops from seeding blight and root rot, and affects most crops. And it goes all the way to fusarium head blight affecting, of course, most cereals in different degrees. So root rot in cereals is also a disease complex, and it's more severe under dry and high temperature conditions, and thus we have been seeing it more often in the last few years because of that. And it's often what we call an invisible disease, which robs wheat and barley of yield and quality. And the main root rot pathogen in cereals, which is Coccocleobulus sativus, or we usually refer to it as the CRR pathogen, can persist in soil for a very long time. And the reason for that is that it's thick-walled and melanized spores, which are quite resistant to decomposition. And when infections are caused by fusarium species, and usually they are, it's a combo and a complex, the fusarium colonization of roots allows the survival and carryover of fusarium pathogens that can cause fusarium head blight, because in the same pathogen that causes fusarium head blight can also cause root rot. And among the different species, durum wheat is the most susceptible. And just a couple of pictures. Unfortunately, there hasn't been any information on root rot susceptibility in wheat in the varieties of grain crops publication, and this has been the case for a few decades already. It's not, unfortunately, it's not root rot in cereals is not considered a priority disease, or hasn't been considered. A few years ago, we published on differences in root rot and fungal populations among different cereals, and this is under organic management. And in addition to that, we have other publications on the impact of agronomic practices on root rot and the different species colonizing roots. So compared to, as I said, up until now, all the studies that we have done under organic and non-organic conditions, were in regards to rotations in the same area in current and semi-arid environment. So compared to those studies, we found that there were significant changes in fungal communities in the intercrops and in the soil during wheat the following year. And unfortunately, I don't have time to elaborate on any of this. We're in the process of publishing this information. And we found that in contrast to previous studies, Fusarium species were the predominant fungi in both the intercrops and during wheat. And I can't go over the reason for that, but that is what we have found. And there were about 20 different Fusarium species that we found in both the intercrops and the following during wheat. So I'm just going to refer to some partial fungal data. So, and in terms of the Fusarium species that we isolated, there was a range in the different species. They range from being pathogenic on the same crops to being weak pathogens and saprophytes. But in addition, some of them have biocontrol potential. In addition, there were also other biocontrol fungi that were present, which have been shown to control diseases and other pests in different crops. Some of these, such as Clonostatis species, and you might have heard about Clonostatis rosea because there has been quite a number of articles from colleagues that have looked into that before. These other species are known endophytes, which are able to also promote plant growth and drought tolerance. And endophytes are organisms that live within plants for at least part of the life cycle. And again, there is more and more information on this kind of organisms lately than previously. So not all, we found that not all intercrops had the same impact on root rot severity and fungal populations. And this is going back to phase 1. And from 17 and 18, we found that in the field pea oat intercrop, we found similar or higher root rot in field pea intercrop with oat than in the monocrop. And this is related to the fact that field pea and oat share similar Fusarium pathogens. So therefore, there was an increase of root rot caused by this organisms. There was a synergistic effect of these different species. But on the other hand, when we looked at lentil and mustard, there was less root rot in lentils when they were intercrop with mustard than in the lentil monocrop. And this could be attributed mostly to allelopathy from the mustard and or an increase in biocontrol fungi, among other factors. And this is an area that we were, of course, very interested in. And also we had been looking at this kind of biocontrol potential of different organisms controlling diseases and legumes. And in regards to this phase 1 effects, there was we had an article published in Top Crop Manager in 2021 that you might want to take a look at. It has more information on it. In terms of phase 2, which is, as you know, the durum wheat grown after the intercrops, we found that the effects of the intercrops on root health and fungal communities were also carried over to the following year. And we found significant differences between the two intercrop combinations in root rot and the levels of Fusarium and other species in how they manifested themselves in the following soil during wheat crop. And again, there's going to be an article coming up this summer on the impacts of intercrops on root rot and the health of the durum wheat following them. So I just selected 3 of the main pathogenic Fusarium species that we found, that we isolated from durum wheat. And Fusarium avinesion, you might be familiar with it. It's one of the main Fusarium head blight pathogens. found in Saskatchewan. As you can see, it was present at the highest level in forage pea manure, but it was present at lower levels in all the other crops, including the monocrops. Oxysporum and Solana, which are the other ones that I selected, are almost exclusively pathogens of legumes. Also, they're also found in cereals. And as you can see here, the difference in levels were attributed to the previous mustard and lentil mustard intercrops. Here, in terms of the severity of root rot, the lowest levels were found. Root rot in durum wheat were found following mustard, and the intercrops with mustard at the highest ratio. And the lowest levels of all three pathogens were also found after mustard, and also in some cases after some of the intercrops with mustard. So there was it was quite significant difference that we found, and it was quite striking. So among the fungi with biocontrol potential, as I indicated before, some of the Fusarium species have biocontrol potential, and the one that we isolated the most and we are going to address here is Fusarium equisetae, which is a very common soil fungus, and also Clonostachys species, which I also mentioned before. So this is the same data, the kind of data in terms of the isolation of this fungi, biocontrol fungi after growing intercrops. And so we have equisetae and Clonostachys species. I should mention that most of the Clonostachys species are Clonostachys rosea, but there are also other species as well that that we included here. So contrary to what we observed in relation to the abundance of Fusarium pathogens, here you can see that the highest levels in equisetae and Clonostachys species were found in after mustard, and some of the lentil and mustard intercrops, especially the ones where mustard was present at its highest level. So for those of you who are not familiar with principal component analysis, I don't want to give you a headache, but this is actually part of one of our manuscripts that we're working on. But principal component analysis is a technique that is commonly used to explore different associations among different organisms, and associations between, in this case, between fungi and root rot levels. And all I wanted to point out, this includes more fungi than what I presented in the previous tables, but not all of the fungi that we are isolated. These are the most common one. So here you can see the green, the light green, and the dark green corresponds to oat and the pea oat intercrop. And as you can see, they are closely associated with root rot levels, so they're closest to it. And but on the opposite end, you see the red and the I think this is kind of like a brown color or dark red. These are mustard and the lentil mustard intercrops. And here you can see, I'm not sure how well you can see here, this is Clonostachys here and Fusarium equisetae. So you and the other fungi here are Fusarium species and other species as well, which are mostly associated with mustard, but are not usually found in other crops. So as you can see, mustard and the intercrops mustard associated. Kind of opposite from pea oat and clearly associated with biocontrol fungi. So this is the what I presented before in terms of the positive and negative impacts of intercrops on the following year. And again, I wanted to emphasize that we're talking about very dry weather that we had not only in 2021 but also 17 and 18. So among the negative impacts, we can now add a similar or higher percentage of fusarium and root rot after the field pea oat than its monocrops and forage pea manure. So that was what we consider a negative impact. Another positive impact is the lower level of root rot and higher levels of biocontrol and growth promoter fungi after mustard and its intercrops, at least its intercrops with the highest level of mustard. So, and we were very interested in this, not only because we had been looking at some of these organisms, but also there have been publications in the past. This one is, and there are many publications. I just selected two. This is in regards to Fusarium equisetae in terms of its biocontrol potential. And here, this is one from Ontario. And as I said, there are many on Clonostacis rosea as being a biocontrol organisms and also as an inoculant to enhance growth and productivity of crops. And it has been promoted and usually used for higher value crops and greenhouse crops mostly. So I just as I indicated before, we have been looking at this for a number of years, and this was also the subject of a current project under organic cluster, and this has been funded by the Alberta Wheat Commission, PHS Organics, and Sask Wheat, and we have been examining the impact of introducing biocontrol organisms to different pathogens, particularly Fusarium species, but also other pathogens. So this is an article in Top Crop Manager regarding this work which came out in 2020, and which has published this study as well on the mode of action of different biocontrol, including Clonostacis species, on Fusarium species, and also Cochleobolus sativus, which you might remember is the main root rot pathogen in cereals. And these are all pictures from the microscope, and we examined the mode of action in terms of the parasitism of by all this biocontrol fungi. And we also conducted some greenhouse studies, growth chamber studies. And here you can see the CRR pathogen on the left by itself, and here on the right when it was seeded with a Clonostacis species. This one is the same. For this is infected with Fusarium avinesim, which, as I indicated, is one of the main Fusarium head blight pathogens, and also one of the main Fusarium root rot pathogens. And here on the right, you see better growth when you introduce Clonostacis species. We have also been conducting field studies on this, but unfortunately, due to the drought that we have been having, the results haven't been that great yet, and they had to be watered by hand every day in 2021. So we're always hoping to for better environmental conditions to take this to the field. So following the lentil mustard intercrops, then the lower Fusarium root rot that we observed in during wheat could be due to the higher levels of biocontrol organisms, which we showed in the lab and in the growth chamber, that they do control, they are able to control Fusarium species. It could also be related to allelopathy from mustard, and as you probably know, mustard is one of the most allelopathic species there are. It could also be due to lower susceptibility of mustard to Fusarium pathogens. and efficient pathogens of other crops. And as I indicated, mustard has its own fusarium pathogens, which are not really very pathogenic. They are present in mustard roots, but they are not. They don't have a high pathogenicity on it, and there could also be different synergistic effects and other mechanisms being involved. And of course, we are interested continue being interested in the biocontrol organism. So, significant findings such as this might only be identified and quantified under organic management for obvious reasons. When you introduce fungicides or other synthetic chemicals, it could affect, especially fungicides, it could decrease the levels of these organisms, particularly Clonostatis. And there's fungi, biocontrol fungi have indeed been shown to be more abundant in organic than non-organic systems. In terms of Clonostatis, this is a publication from Europe, and we found similar impact of organic versus non-organic in regards to fusarium equicity, that we published in 2011. So, and there are various strains of Clonostatis rosea that have been patented for biocontrol in different countries, and you might have seen some articles. There have been articles on these organisms here in Canada. But as far as we know, so far, there are no successful products with this biocontrol species or genus, because there are other species within the Clonostatis genus. And so no product has been commercialized in Canada. And we have been interacting with a company in Ontario that has been trying to commercialize these organisms, but there have been problems in terms of regulatory issues, I guess. So given that in addition to the reported ability to control crop diseases, endophytes such as Clonostatis species and others may increase crop performance by promoting plant growth and improving drought tolerance. The question is, how can we use mustard or other brassicas in intercrops or crop sequences to promote increases of this fungi? So in other words, can producers take advantage of these findings? So producers might be able to, based on our research, producers might be able to naturally increase Clonostatis rosea and other related species. Our intention is not to commercialize any of the biocontrol organisms that we have identified in this project or in other trials, and the other trials actually include cover cropping trials that we have through different projects, and where we are making the same observation in regards to other brassicas that we are using as cover crops. So promoting its natural presence might provide disease control and promote crop growth and improve drought tolerance as well. However, as I indicated, the negative impact of mustard and its intercrops when the companion legume grows poorly due to dry weather, we don't know whether this would be the case under more suitable conditions for legume growth. Well, what I presented was from dry years when legumes didn't grow well. So the negative impact of mustard on soil nitrogen and the resulting growth of the following crop is of importance, mostly under organic and low input management where no synthetic fertilizers can be used. So how can we remediate the negative aspects of a crop species, which might help to increase biocontrol organisms and also provide increased crop growth and drought tolerance? So it would be expected that a legume, especially when we use it as green manure, would help restore soil nitrogen after growing a brassica or its intercrops at high ratios, especially when the companion legume grows poorly. And when the legume grows poorly, we expect that the negative impact on nitrogen, of course, is going to be the greatest. However, sorry, I missed an H there. However, the big question is whether brassicas have an impact on nodulation and or mycorrhizal fungi in the following legume. And this is the subject of our, sorry?
Sorry, you got 2 minutes.
Oh, sorry. And so this is the subject of our current and upcoming research. So yeah, this is I only have a couple of slides. So when designing crop intercrops, we need to consider the trade-offs, take advantage of the positive attributes and remediate any negative impacts. And we need flexibility also, depending on factors such as environment, disease levels, and soil nitrogen, in addition to cropping systems. So the design of this cropping sequences and intercrops would be different. I'm not going to be talking about this. This is so I just wanted to end by showing a picture of our first field day in Swift Current in 1921, which we just commemorated 100 years in this past year. So it was quite a finding when we found this picture.
Oh,
Anyways,
that's definitely very interesting.
yeah.
Okay.
Actually, I wanted to. I talked to the museum about it, but we haven't really gone too far in that.
Okay, thank you very much. We have time for a couple quick questions. The first one was, what was your row spacing in alternative rows?
Yeah, and 21 was, I believe it was 8 inches. Yeah.
Thank you. The other, do you have a recommendation on a seeding rotation when using intercrops? Are there any changes you would recommend?
For the intercrops or for the rotation?
Intercrops.
For the intercrops, yeah. We will continue looking at field pea and oats. We think that is it's an interesting combination, and there are some pros and cons of using that. We also plan to continue looking at lentil and barley. And the other one that you might remember that we looked at chickling vetch and mustard this year. We are not growing the chickling vetches for grain, of course, but we and I don't have any data yet to show you, but we are using it mostly as a leaven mulch, and with the and it doesn't really. I mean, it starts to flower after a while under the mustard, but it doesn't produce weeds. And so we are looking at the contribution of the chickling vetch with the mustard for nitrogen.
The long-term rotation study, which is Canada's oldest organic versus conventional farming systems experiment, which has completed 28 years. Together with colleagues, Martin founded the U of M's Natural Systems Agriculture program, which explores cropping systems based on processes found in nature, specifically the natural grassland ecosystem of Prairie Canada. So we'll start whenever you're ready, Martin.
Okay, can you hear me and see my slides?
Yes, we can hear you and see your slides.
Okay. Okay, great. Good morning, good afternoon, wherever you are. So thank you for inviting me. I'm Martin Ents. I'm here in Winnipeg in Treaty 1 territory, and I'm going to talk to you about some of our work. I really enjoyed the morning. I've been able to sit in the whole time. I finished teaching at, I guess, 9 o'clock your time, and so it's been a great morning. So intercropping and organic farming, I am just a dirt kicking agronomist. I don't have the skills that Miriam has to look at all the pathogens, and I'm really jealous that I don't get to do my work closer to where she is. So thanks to the graduate students who've worked on this project over the years. And the outline of my talk is I want to highlight green manure cover crops and the role of intercropping there, late season cover crops, and then grain intercrops. And I've shown you the picture of the other air seeder that organic farmers can sometimes benefit from. So let's get going. When we look at green manures, you know, those crops that we grow for sometimes for a whole growing season to enrich the soil, to bring nitrogen into the organic system. There are many different species to choose from. And there is a green manure for almost any situation in Canada. If you're really dry, something like chickling vetch or maybe lentil. If you're really wet, something like faba bean, or if it's really hot, then something like soybean, perhaps mixed with warm season grass. And so these are all the types of green manures that we've looked at over the years. But the question that you're interested in is, you know, what happens when we grow them in mixtures? And I agree, that is an important question. And in fact, that is the best way to grow a green manure. Pea oat mixtures are very popular. I'm now learning that they're helping promote fusarium, but we've had a lot of very, very good success with these fully leafed peas and oats. And they are widely used by organic farmers. In some cases, especially when it's wet, and in the wet parts of the black soil zone where I'm located, we think about introducing other crops into that mixture. So for example, faba bean. And this is a really good idea to spread out the risk. Also gives us a chance to introduce what is among the best, although sometimes unreliable legume into the system, and that's faba bean. And it also helps us deal with situations like you can see in this picture where the farmer was not able to get any leafed peas. So they're growing semi-leafless peas. And semi-leafless peas are great for grain production, but they're not ideal for green manures because they allow weeds to creep in. So the mixtures in the green manure system are really important. And now with the interest in cover crops growing, we know that mixtures, sometimes very simple mixtures of a grass, of a legume, maybe a brassica, maybe a warm season grass in addition to a cool season grass, they can really help organic agriculture grow better green manures that suppress weeds. So what I want to share here is a study that we conducted recently where we looked at the biomass of green manures on 41 different fields in the eastern prairies. So we're looking at sort of halfway through Saskatchewan and then east and including Manitoba. And we were just interested in, you know, what was the proportion of the legume versus the weeds versus the other plants that were maybe grown as grass companions to these green manures. And this is what we found. This is the data from the 35 fields that did not contain hairy vetch. And you can see the legume on the left, we have the total biomass, which is similar to pounds per acre, and you basically multiply that by 2.5%, and that gives you how much nitrogen you've contributed. And you can see the amount of legume really, really varies, and it tends to be low. And that's one of the challenges with our green manures right now, is they're often phosphorus deficient and the legumes are not growing as well as they could. And so from this, we've learned that adding phosphorus is really important, and adding phosphorus to the legume phase, to the green manure phase, is an excellent place to add, whether it's manure or compost or some other nitrogen phosphorus source that's not rock phosphate. And then we can see the weed biomass is actually quite small, and that really surprised us in this work. But anyways, this is what we're seeing. We are seeing mixtures as green manures, and then the gray bars are the grasses that, in some cases, it may be quackgrass that creeped in. So that's what we found. And based on that data, we went, well, you know, that's not really, the green manures that we're seeing on organic farms are, you know, there's some room for improvement. I mean, some farmers are doing an excellent job, but lots of times we're not getting the biomass and certainly the nitrogen that we need. Are there other plants that we could think about in that mixture? And so we started playing around with hairy vetch, and this is just a picture of the hairy vetch plant and its nodules. And when we looked at the fields in this survey that had hairy vetch in them, and the hairy vetch was grown with barley in this system, which I'll explain in a moment, we found massive differences in the productivity. We found legume biomass that could produce, you know, well over 100 pounds of fixed nitrogen, and that's going to allow you to grow very big crops. And so let me tell you a little bit about the system, which was really developed by my former field technician, Keith Bamford, who, you know, we were growing all kinds of different intercrops and cover crops. And so we grew hairy vetch with barley and oats and other things. And then we cut the barley for silage, and that was our idea. We're going to cut it for silage and let the hairy vetch regrow. But we never got really good regrowth on hairy vetch. And so we started blade rolling it. And yes, this was a 1-year green manure, so we gave up a whole year of production to do this. And then we zero-tilled our organic crops into that mulch the following year. And certainly in much of the black soil zone, the hairy vetch winter kills. It winter kills, we've done this probably 14 years, and it's winter killed every year where we are. And so here we have organic wheat, no-till seeded with an offset no-till disc drill into this mulch. The combination or the intercrop of these hairy vetch barley really serve an important purpose. So here you can see the hairy vetch barley mulch that's winter killed. We've no-till seeded flax into there. And if we pull the mulch apart, we can see the barley is actually separating the soil from the nitrogen-rich vetch. And that is because when we blade roll it, we kill the barley, but we don't kill the vetch. And the vetch is a second half team. It grows right till freeze up. And that's when it produces most of its biomass. And so we ended up with this very nitrogen-rich vetch separated from the soil by this barley, which makes it slow release. If you put that nitrogen-rich vetch right on the grass, right on the soil, it'll break down much quicker. And that's what Carolyn All did her PhD on. So that's a little bit about you know, mixtures. And then one other thing is that organic farming, organic farmers really manage decomposition. And it's just like cattle farmers. They're managing decomposition in the rumen. Organic farmers are managing decomposition in the soil. And when you have a monoculture green manure, that decomposes very differently than if you have an intercrop that might have some grasses and some fiber in it. But you want the nitrogen. So that's just something that I'd like you to think about. The other thing that came to mind as Miriam was talking was we have so much mustard always growing in our organic systems. I wonder how much that has helped us manage diseases over the years. Okay, the next system is the late season cover cropping. Now, this is not for everyone because we don't always have the moisture and growing resources after we harvest our main crop. But there are successful systems out there. And one of the ones that we have found, we've compared two systems. One is putting red clover or sweet clover or alfalfa in with the spring crop, seeding it at the same time. And we've compared that with planting peas or chickling vetch or lentil after we harvest the spring crop. Which one is better? Well, in our experience, the red clover, the clover seeded at the same time as the cereal gives us a slightly higher biomass over a number of different site years. And we've published that work. So I can send you the papers if you wish. And one reason why the small seeded legume is successful is we're seeding it at a time of year when there's moisture. And sweet clover, for example, can be seeded quite deep. So people had asked about seeding depth. And so this is one thing that can help us, add some nutrients. And I like the idea of mixtures. This is where the intercrops come in, of legumes, where it's not just alfalfa by itself or a late season green manure or red clover, but a mixture of the two. And we're talking here about systems where we grow these, let them provide some ground cover in the fall, and then get rid of them and then grow another cash crop the following year. This is where the air seeder comes in. This is the most sophisticated cover crop seeder I've ever seen. It's on a lily harrow or Einbeck harrow in Quebec. And when they harrow their wheat, the last time they blow on their clover. And these types of systems can also work, especially in much of the black soil zone. And then I thought I would mention that when we grow peas for green manures, pea oats or pea barley or something, we used to always terminate the pea at flowering time because that's what the textbook said. And then we said, well, we ran into an organic potato farmer and they planted a second cover crop after their first one. And so what we started doing is just letting our peas form pods and then killing them. And then we'd get a volunteer crop. So we'd get a second catch crop for free. And finally, I want to talk about grain intercropping. And we haven't done tons and tons of work on this, but we've done some. I had a graduate student work in 2004 and 2005. So Jackie looked at a whole bunch of intercrops. Here on the left is the hairy vetch wheat in 2004 and here it is in 2005 when we were in a wetter situation and we had less nitrogen. And what we learned is hairy vetch is very dangerous to grow with wheat. It just takes over. On the bottom, we have flax wheat on the left in 2004, and then on 2005, we made a seeding mistake and had way too much flax. But I really love this intercrop, and I have heard of organic farmers growing. Whenever they grow wheat, just throw some flax in. Now with the gluten intolerance, the flax buyers are getting fussy. It's unfortunate because it's a very nice way to grow flax. So what did we learn in this study where Jackie looked at, I'll list all the treatments. We had wheat and oats, wheat and barley, wheat and spring rye, wheat and flax, wheat and pea, wheat and mustard, wheat and a number of cover crops, red clover, hairy vetch, annual ryegrass. And all of those were grown at a half rate of wheat. And then we also had a half rate of wheat, seeding rate of wheat by itself, and a full rate of seeding of wheat by itself. So we did this over 3 site years on organic farms. And what we noticed is that the biggest difference is that when we reduced our wheat seeding rate, we reduced our leaf diseases. So this was % of leaf area that's infected by a pathogen. Same thing in all 3 years. But of course, we don't want to just grow a half rate of wheat. We want to grow an intercrop. And so the intercrops, especially things like wheat pea, did reduce our leaf diseases. When we look at weeds, if we look at the full rate and half rate of wheat, we can see over here, we've got weed biomass in 2004, 2005. We can always see a significant increase in weed biomass at harvest when we reduce our wheat seeding rate. So if we were trying to reduce our leaf diseases by reducing our wheat seeding rate, that would work, but we'd have more weeds. So that's where, of course, intercropping comes in. And what we noticed is the intercrops were giving us, some of them were giving us much lower weed biomass than growing growing the half rate of wheat. So by putting an intercrop in there, we filled the space that the weeds were taking over, and that, I mean, that's not rocket science. But then if we look at the economics, they were never really very positive. And so here we have the full rate of wheat, and this is the net return per hectare. So you can see at the bottom here, these always gave us the highest net return, except in 1 year, the mustard wheat rotation was statistically significantly more profitable than the full rate of wheat. Otherwise, intercropping was rarely more profitable than a full rate of spring wheat. But there were those other benefits like weed management and also disease management, especially disease management. And where we are in the Eastern Prairies, leaf diseases are a major concern. More recently, I had a graduate student look at pea intercrops with oats, barley, and mustard. And these are some of the economics, because I will show the economics. And we looked at the seed costs. We looked at everything from the seed separation costs, and we looked at the market prices for the crops. And this is published now in the Canadian Journal of Plant Science. And we noticed that, the wheat, as we would go with, we grew peas by themselves, and then peas with a low rate of barley, medium rate, high rate, or with oats. And we always noticed a reduction in weeds. So the intercrop consistently, especially as we increase the rate of the non-pea, the weed control benefits increased. I should add, we used a full rate of peas in here. So we were interested in growing organic peas for the protein market, but the question we asked is, if we added a little bit of these crops to those peas, would that improve the production? And the cereals, the oats and the barley, always improve weed control, even with a quarter of the commercial seeding rate. The mustard was inconsistent because of flea beetles. Here are the economic returns from that. I'm going to draw your attention just to these data here, where we look at the pea monocrop at the full seeding rate, the intercrop at the low seeding, medium seeding, and high seeding rate. When there's a bracket around the number, that means it's a net loss. Now, what we found is that pea barley, as we added barley, especially the medium seeding rate, we lost less money. And the economist on the committee, Derek Bruin, said, even though these numbers aren't statistically significant, he said, this is noteworthy, where the intercrop was improving the economic situation. If we look at pea oat, we actually had the same non-significant increase in profitability. If we look at pea mustard, this was a real unfortunate situation because we were in very, very dry years, and we had a lot of flea beetles, and the flea beetles didn't start eating the mustard until the canola got swathed in the region. And then so we grew the mustard, suppressed the pea yield, but we never harvested much mustard seed. So that's the circumstance here. So I wouldn't give up on these mustard intercrops, but I want to explain why, the more mustard we added, the more money we lost. So just before I go, a couple of other things. Jeff Shano did a really good job talking about nitrogen. I want to talk a little bit about phosphorus, because this is an issue in organic farming, and this shows the rhizosphere of, on the left, we have faba bean, on the right, we have corn, and in the middle, we have soybean. And this is showing how these crops affect the pH of the soil right around the roots. And so we can see that something like faba bean creates an acid condition which allows the faba bean to solubilize soil phosphorus or phosphorus from the manure. And we see that maize is not very good at it at all. And I think oats is probably about the same as this, but I'm just speculating. So when we think about intercrops, phosphorus also becomes interesting. So here we have an intercrop in northwest China, and you've got maize growing with chickpea. And when I think about this picture, then I go, oh, I wonder if that chickpea is helping that maize access phosphorus. And if we look at the data, and a lot of really fantastic intercropping data comes from China, at least insofar as nutrients are concerned, that's exactly what they found, that the over yielding, which means that higher leaf area or land equivalent ratio that previous speakers have talked about, was really something they observed when faba was used on a low phosphorus soil. So that's interesting. Okay, just finish off with a couple of other intercropping stories that are a little unconventional, but very important for organic farmers. One is using trap crops. So this is an organic flax crop, and putting peas around the edge can help suppress the movement of grasshoppers coming in. So that's really a form of intercropping. So that's a system that can work. Here's a system that doesn't work, but I'll tell you about it. So Keith was very interested in suppressing weeds within the row of organic soybeans. So we grew a very light rate of rye planted at the same time as the soybeans. And well, it helped suppress the weeds. But over here, where we didn't add the rye and we had more weeds, we had a much higher yield. So some things work and some things don't. But the whole idea of using rye as an intercrop in organic production, together with things like blade rollers or even flail mowers, is growing, and there is a lot of potential for various applications of rye because of its allelo benefits in organic pulse production. And I'll just end off with this image and then go to my conclusion slide. This is one of my first intercrop systems that I ever saw, which was in Denmark, where they had gone into organic forage grass seed crops, and they'd taken a back-swept knife and cut some of the grass out of there and put clover in as a way of feeding nitrogen into the system. And it worked. And I was quite surprised, actually. And I think this is the kind of thinking that we need to apply to our organic production, even grass seed. So in conclusion, intercropping in organic farming. Well, I will say that intercropping in the green manure or the cover crop phase is definitely very positive. We've seen some significant benefits to doing that. And then think full season, especially in the black soil zone, where we never know when we're going to have a wet fall. So we might as well prepare to make some nitrogen beyond the 25th of July. When it comes to grain intercropping, we know that it reduces leaf diseases. We know it reduces weeds. Yields and net returns are really not. We haven't seen compelling data for that. But in an organic system, it might be worth it just for the weed and disease control. But then the other benefits that could be the quality of the crop, the just no dirt tag on the pulses. I mean, that is worth a lot. And then I would say that Saskatchewan is so lucky to have the organic program that Miriam leads at Swift Current. I really mean that, because keep funding it, because there's all kinds of important data coming out of there, and you're getting tremendous service from Agriculture Canada through that. So that's it for me. Thanks a lot for your attention.
Thank you very much, Martin, for that great presentation. As a reminder, if you have any questions, please enter them into the chat box in the bottom of your control panel. The first question we have is faba bean canola intercrop a good option for the moist black soil zone?
Faba bean canola intercrop. I've never done anything with faba bean and canola, and I don't work with canola because faba bean, like when I worked in Australia, they called it failure bean for a reason. It's a bit inconsistent, and that's my only concern. I like the idea of peas with canola better than faba bean with peas, with canola, but limited experience.
Okay, thank you. Maybe a small on-farm trial would be useful. And I also was wondering, do you know if there's any research on acidification for rhizospheres for other pulses that are available, like lentils and chickpeas?
I think most legumes do some acidification because of the nature of the nitrogen fixation process. But obviously the soybean and faba bean were very different in the example that I gave. I cannot answer that question. I can try to find the answer and I will let Sask Agriculture know. And that's a very, very good question. I should have known the answer, but I'm not going to tell you something I don't really know.
No, I definitely appreciate that. And you can't know everything, so that's perfectly fine. What rate do you recommend for wheat flax intercropping?
Well, I was excited about wheat flax intercropping, and then farmers were telling me that, the buyers of wheat don't want, or the buyers of flax don't want wheat chips or even wheat dust in it because it could contain gluten. So be very careful. What we've done is I was talking to the Horkoffs actually in Kamsack, and they were saying that just you might want to talk to them about rates. I think the rates can be quite low. It's just maybe a third rate of flax, but you're not getting a lot of flax out of the crop. But it does grow with wheat, and it is flax by itself can be very weedy, and so that's what I'll say about that.
Okay, thank you very much. That's all the questions we have. So thank you again, Martin, for all the great information.